mitigation is, again, measured by whether you see disease or not. That's success. Mm-hmm. You have no idea whether your mitigant actually inactivated the virus itself. Because the virus is a structure, right? Are you destroying the structure or not? And more often than not, the experiments we run are limited by our understanding of the effect of the mitigant on the virus itself.
This is the Feed Science Podcast. On behalf of Wisenetics, I'm Adam Fairnolds from North Carolina State University's Prestige Department of Poultry Science and the feed milling program here. My guest today is Dr. Declan Schroeder. Dr. Schroeder is an associate professor in the Department of Veterinary Population Medicine at the University of Minnesota and is going to talk to us a little bit about possible transmission and testing that would relate to swine virus potential as it might move through the ag system and potentially even through the feed system. So with that, welcome, Dr. Schroeder. Thanks for joining us.
Oh, thanks for having me, Adam.
You bet. If you wouldn't mind, give the audience a bit of a background on kind of your background as to how you got to be what you are doing today, and then also what those things are, what your current focuses are, and how they might relate to what's going on in the feed industry.
Right. So I think the best way to describe me is essentially thinking of someone as a virologist. And in fact, I'm more of a general virologist. I've always had an interest in viruses and why they are here, where they occur naturally, what's the ecology. And that's the key word, ecology, because most often we think of viruses as purely as a pest and a pathogen and trying to get rid of it, thinking that it's an alien. So my interest is really about, it's a natural system. Can we understand it? If we understand it, then how best can we deal with it? How can we live alongside viruses? So that's my interest, and as a result, I'm a molecular virologist. I've worked on a variety of viruses. I was actually brought up on plant viruses, but I quickly went into algal viruses, which some people might think is crazy. Why would algae be important? But in the end, algal viruses, we discovered lots of viruses that were very similar models to animal viruses, not plants. And that transitioned my interest into honeybee research, honeybee viruses. Okay. And then finally, I moved to the University of Minnesota about 5 years ago, where I expanded my research even more, looking at viruses in swine, in dairy cattle. But more interesting, really understanding what viruses are in terms of their structures, how best to study them, especially in the industry of food production. And most recently, I've just acquired some work on even influenza viruses. So yeah, that's my interest is viruses as a whole.
Okay.
Eastman serves veterinarians and nutritionists in agrochemical and animal health industries by helping them select, evaluate, and implement innovative nutritional programs. Eastman works with your team to customize your gut health approach in feed and water. Eastman's approach addresses nutritional and bacterial challenges and finds new ingredient preservation and hygiene solutions. Explore ways to accelerate and innovate your programs. Contact the animal nutrition team at eastman.com.
To start off completely out of left field, because you said something that is now interesting to me, before we get in the animal side, why would algae type viruses be more related to plant viruses or more related to animal viruses than they were to plant? That's an interesting thing. I wouldn't have thought that either. Right.
So this is if you take the mindset of the host, like the animal, the plant, algae, you would say by just familiarity, you would say, well, plants and algae, they both photosynthesize and they both capture carbon to photosynthesize to produce oxygen. So they must be related. And actually, when you go into the genetics and you understand the evolution of how these systems evolved, we find that algae actually evolved very early on prior to both animal and plant evolution. And actually, a lot of the processes, a lot of the membranes, a lot of the way in which we move things across membranes are actually algal-like. And a lot of these things have been founded in algae first before plants. And so when you look at the genomics, you look at the way the cell evolves and how it interacts, we found they're actually very similar. And then when we then discovered the viruses alongside it, we realized actually the viruses use an animal-like infection strategy rather than a plant-like infection strategy. So plants require a lot of vectors, a lot of manual manipulation for viruses to be transmitted. Whereas in animals, yes, we have vectors, but there's animal-to-animal transmission. And there's a lot of that cell-to-cell transmission that we get within algal viruses. So the more we start to study these viruses, and actually one of the algal viruses that I helped discover 20 years ago was a virus that's most closely related to African swine fever virus.
Interesting. That's interesting. Okay, so before we get into the really scary African swine fever, and you mentioned high path avian influenza as well, how about on viruses so far that we've seen in a lot of it's been in the swine industry? We think about things like PDV or things of that nature that we've already had to deal with. What have been the things from a virology standpoint that have made those things easily transmissible, but also maybe somewhat, I'm not going to say easy to control, but in some ways controllable by some of the strategies that we have used?
Right. So, when animal scientists or veterinarians come to me as a virologist, they want general questions answered, right? They want to know, come on, Declan, tell me, how do I work with this virus? What kind of general answers? And this is actually the same for the industry, whether it's swine, poultry, whatever. It really doesn't matter. What we're trying to do is characterize viruses by their properties and say, well, if you have this virus and we know belongs to this family of viruses, then we have some sort of understanding of the likelihood of these viruses causing harm. Now, within the swine industry, if you look at the diversity of viruses in swine, they are dominated by RNA viruses. Mm-hmm. And actually, even within the RNA virus group, you have those which are either retrovirus of origin, which becomes a DNA, or you have a positive stranded RNA virus, which is like PERS-like, or coronavirus like PDV, or you have the negative stranded RNA viruses like that of influenza. And then what we try and do is we put all these viruses together, and you ask, well, the RNA viruses, they have this sort of shape, they have this sort of structure. So as a result, based on the commonality, we say, well, actually, this is the potential for them to survive in nature, or survive in feed, or survive on surfaces and in fomites, right? So that's where we try to generalize. The problem lies is when we start to dig deep, we start to realize, yes, there's lots of generalities we can use, but also you actually then realize you're limited by the knowledge you've gained on that system. So to answer your question, when PDV came in, for example, the only explanation that was derived in terms of the origin of this virus was through feed, right? It was never proven. It's very hard to prove something after the fact, right? Because we weren't monitoring. And that's actually a lesson we should think about now for future foreign animal diseases. We don't really monitor and have a surveillance program holistic enough to be certain of the timing when something comes in. But with PDV, when PDV came in, the only possible conclusion could have been, well, it must be because the virus was in the feed. And then various studies were carried out, especially led by Scott Dean, who was first at the University of Minnesota, and then he continued his work in Pipestone. They essentially found that a lot of these viruses were stable, right? And so then you start thinking, well, if PDV, which is an RNA virus, is stable, does it mean all the RNA viruses are stable? And actually the answer was no. These viruses have unique properties that make one stable versus the other not. For the RNA viruses, we have no idea why. There's still no understanding, a mechanistic understanding why PDV, say, is better than PERS. Although there's a recent study that suggests that PERS could have been moved and transmitted from one herd to another through feed. The mechanism behind that is very different, very difficult to understand. But what's key, Adam, is that when one understands the stigma, and especially in swine, we know a lot about the RNA viruses. What we know less about is the double-stranded DNA virus. And the only example that is ASF, right? Mm-hmm. So then you try and begin, they ask me, so what's my prediction? How would we understand? Well, actually, we don't. We don't have a good prediction. We don't have model organisms in terms of model viruses to compare and contrast against, right? We know a little bit about RNA viruses and their stability profile, but we know nothing about double-stranded DNA viruses. And we're starting to understand a little bit more about the single-stranded DNA viruses. But yeah, people are looking for general answers, and the answer is we need to understand better models. We need to get better models to understand stability, especially in feed.
Sure. Okay. So from the virologist perspective, and so we've in different times when we've talked about things like PERS and PDV, we've talked a lot about how we might mitigate issues through management and biosecurity protocols and things like that. And a lot of those kind of tend to make some common sense from the actual virology perspective and how the virus works and transmits and replicates, all that sort of thing. What are the strategies that have worked or have made the most sense in those mRNA type viruses in order to hopefully not either have it in the feed, or if it horribly is there, what we do about it?
Right. Great question. So it's a really hard question to answer because it's actually, well, let me step back for a second. So when trying to work with viruses, one is limited in terms of what you can do. And actually, when one thinks about mitigation strategies or thinks about how we go about addressing this question, how you go about testing it, you are almost more often not reliant on having a system that you can test, right? A cell culture system, an animal system. And so when you look at viruses, and especially when you're trying to understand feed and whether the virus can be transmitted from feed to an animal system, you are constrained by what you can do in the lab, right? And so the tests we run, there's all tests to do with whether a virus in the presence of a mitigant can cause infection in a cell culture, but your unit of measure is infection. And it's not actually a unit of virus measurement. If you think about it, you're not measuring the virus, you're measuring the biological activity as a result of what viruses do. They cause an infection. Mm-hmm. And so a lot of the problems we have with the way in which we interpret our data, especially when it comes to feed data, is because we are limited by the dynamic of the infection cycle, right? And so we're measuring whether that has an effect and what's the cause and how that translates into disease. So let me go back to your question. Knowing that, when we do work on something like feed, for example, and you try and give an animal a feed which has a virus present, you are limited by that small window of you looking for a disease outcome in an experiment design, which is more often not constrained by money and time. And so you look for disease in a short window, right? And then in fact, what you should be trying to do is do transmission studies and actually ask, well, how long does it take for those animals to go positive? And we just can't do those experiments. It's just prohibitive. And so then we go back to cell culture. And in cell cultures, you do these experiments, but then the feed in itself is really messy. If anyone who's ever worked with feed knows how complex a feed mixture is. You put that into cell cultures, the cell cultures behave differently. And so all these sort of questions we have about what's the threat and how do you go about understanding what the virus is doing in the context of feed and an infection is always reliant on these sort of animal-like disease studies, right? And so then you add to that mitigation. Now we know, for example, like I said in the beginning, many RNA viruses are very unstable because they're RNA-based and their properties are very well described. And so in terms of mechanistic understanding of viruses, of feed being a transmitter of RNA viruses, it's still not clear because these viruses should be unstable. Just like coronaviruses, we know when the pandemic hit for SARS-CoV-2, we weren't concerned that much about our mail being contaminated, right?
Right.
The surfaces. So why would we weren't concerned with the food we went shopping if we could go out and get grocery shopping, we would be fine. So the surfaces, the fomites, all these sort of things were not because these RNA viruses. DNA viruses is different. And so then you think about DNA viruses being way more stable because of their very nature. And then they're very more complex. And so we have to look beyond just a sort of pure biological degradation or natural degradation of these viruses. You actually have to apply something. And chemical mitigants is a really good way of doing this. What's also important, I'm going to finish with this, just to finish
Sure.
that thought and tie it all together, is that mitigation is, again, measured by whether you see disease or not. That's success.
Mm-hmm.
You have no idea whether your mitigant actually inactivated the virus itself. Because the virus is a structure, right? Are you destroying the structure or not? And more often than not, the experiments we run are limited by our understanding of the effect of the mitigant on the virus itself. And that's where actually a lot of my work has been going in. As a virologist, I want to go to the virus itself. Can you, and of course, we don't ignore the infection cycle, but understanding what we do and how the virus behaves in feed is not always, you don't always get that information from an infection study. You need to have both. And developing this side of it, the virus particle infection, the dynamic, the particle stability dynamic, and how mitigants play a role there, that is really important. And I think that's where a lot of effort needs to be in.
Sure. Okay. So let me see if I can kind of summarize, make sure I've got this correct. So on some of the things that we know work, again, from like the management perspective, it's common sense of trying not to let, the environment from A get to B, from the farm to the mill, from the mill to the farm or whatever. We know those management strategies. But as far as how the virus is working, or whether a mitigant is working, or whether it's getting to the animal and what it's doing, part of the problem is we have so many different feed matrices depending on ingredients and everything else. We have so many different types of processes, and then we have so many other different things even going on in the management at the farm level that from an experimental standpoint, it becomes very difficult to say, okay, from the virus's perspective, this is what's happening. Is that a fair way to say that?
Right, because we're not measuring anything that's going on in the field. Right. We're getting the end product and putting it in an animal and asking, do I see disease? But it doesn't tell you anything of the process. So in fact, there's no if the assays are not designed to look for what's going on in the process, we are always in a black box. We're always making assumptions. And a lot of our technologies lack that clarity.
So we can do a little bit of, if we do this, we may somewhat know that the end result is that, but why is in many cases still a complete mystery.
Right. Because it doesn't exist. Models don't exist usually. And the way we run our assays, people just not use these assays. We've always reliant on this infection aspect.
Right. Sure. Okay. Excellent. Again, my guest today is Dr. Declan Schroeder, an associate professor and molecular biologist at the University of Minnesota. So, all right, if we've kind of talked about that and you went back and forth a little bit there between the mRNA and some of the DNA viruses. So now if we transition over into these double-stranded DNA type viruses like ASF that has everybody, scared everywhere around the world. Luckily here in the US, we've dodged a bullet so far. You mentioned that they're more stable, and we know that that's going to be a concern as far as what are we going to do about them. What else from them makes them particularly scary as far as what happens if it ended up in the food supply, either ingredients coming into a feed mill or, a contaminated feed mill sending feed out? Well,
the answer to the scary part is that stability part. And actually this goes back to my introduction when we talked about viruses in algae, for example. So one of the things you hear about when you talk about ASF is that ASF is the only double-stranded DNA virus within the family of that virus, right? It's actually the only member of the whole family. Can you imagine how lonely those poor viruses in the family? It has no relatives. And that's actually been correct. Actually, it has relatives. And talking about molecular biology and understanding about evolution, we discovered by the work I was doing in the marine field and algal field, we realized there's a group of viruses that actually encompass multiple families, and one of the families is ASF, the Aspergillus. And actually, we realized that these double-stranded DNA viruses are quite common in aquatic systems, both freshwater and marine water. And these viruses are actually, they infect unicellular. So like algae is unicellular, majority, not all of them. They infect amoeba. And these are large, they're actually called megaviruses because they're large. Simple name, they're giant viruses, right? And so As a result, what we found is these viruses actually live in their environment more than they do in the host cell, because when it infects, it causes lysis, and then it gets released, and it has to find the next cell to infect. So through the life cycle of a giant virus, it occurs mostly in the environment, not in the host. And by understanding that, we then realize that we understand the context within ASF, we're starting to understand that actually there's something about these giant viruses, and not all DNA viruses, but specifically these giant megaviruses, that make them really, really stable. And actually, we have a paper that's currently in review at the moment where we've characterized the structure of this algal virus, and we found that it actually is related to ASF. It has the same complex where it has a double-stranded DNA genome. It has multiple membranes and has multiple capsids, and both of those are shared by this algal virus and ASF. And so to really blow your mind, right? So this family, which belongs to this group of viruses called megaviruses that ASF belongs to, we have evidence to show that you can revive viruses of this group that are over 60,000 years old. So you can have 60,000-year-old sample, and these viruses can be cultured.
Oh, wow.
This algal virus is closely related. 7,000-year-old samples you can pull out from the environment, and the structure and the genome is maintained. And so if you translate that to what and the other virus which ASF is related to is pox viruses. And we know how stable pox viruses are. So the message we're getting back by looking at the whole is that actually ASF is going to be one of the most stable viruses in terms of the swine viruses catalog. Not because it's double-stranded DNA, because it's almost evolved from a common ancestor with all these other funny viruses to be stable in the environment. And so if you understand that mindset from the beginning, you then have to then be careful how you interpret data. So when you run your experiments, you can't just look at false negatives and think, oh, it's fine, I don't get disease, means I must have killed my virus. Well, what have you measured? Have you measured disease, or have you actually measured the virus itself? And the more we get to understanding these viruses, the more you really understand how stable these viruses are. And again, to blow your mind, we did some work and we found that boiling ASF for 20 minutes does not destroy the virus particle.
Great.
Right. Thank you very much. And so this makes you think about, right, when it comes to sort of one of the strategies we use, and we use the poultry as well, we use for swine industry, is depopulation, right? If you have a disease you can't manage, you can't control, you depopulate, you sterilize the system, and then you reintroduce your animals. And this happens over and over again in areas that had ASF outbreaks. And they bring them in, and the virus comes back again. Where does it come from? And so one of the things that's largely been ignored is the environment. And part of that environment is the feed you bring into your environment, right? Mm-hmm. Where does that come from? The feed is exposed to the environment. So it's that double-stranded stability nature of the virus genome, but also the structure of the virus. The virus structure is so complex and so stable.
So how does that potentially change, or I mean, maybe there's only so many things you can do sort of situation, but does any of that understanding of how stable the virus is, how well it can maintain itself in the environment, change things like the more basic biosecurity practices of eliminating movement, and does it force us more into not only are we going to eliminate movement, but we are going to have to do actual, treatment steps, chemical mitigation, whatever. Heat doesn't sound like it's a real good option if you can boil it for 20 minutes. How does that maybe change, knowing more about the virus and how it's different? How does that change practices that to date we say, hey, these work pretty well for things like PDV or PERS or even high path avian influenza that have seemed to be relatively well controlled through depopulation biosecurity, things like that?
Yeah, absolutely. It's like it draws attention to what we thought we knew. And then we think, oh, viruses are all the same. Well, they're not all the same. And then you understand, well, what's the basis for that? And exactly, all the examples you gave are these RNA viruses that don't survive very well outside its host and so on surfaces. Yet we know they can be sources for transmission if your biosecurity is so bad. So our current biosecurity protocols are pretty good, right? So we do find a way, and we do have issues still of endemic viruses breaking in our herds. But it just shows you that we have to reflect and look at our current biosecurity protocols. And if you are reliant on, say, for example, only temperature, that's inappropriate. It's not going to work. And so then you ask the question, well, mitigants, chemical mitigants. Now they also realize the problem is that actually, to be fair, if you think about ASF research has been dominated by vaccine research.
Right.
And because there's only a few labs in the world that can work on it, right? So in fact, the research effort, and rightly so, would be on vaccine development. There's very little work done on stability of ASF, whether it's chemical mitigants or not, right? It's a very small area of field. I think it's probably only like a handful of publications in the literature describing this. So our understanding and knowledge of how mitigants work and interact with these, with ASF is limited. And that's where actually what's quite interesting is that's where a surrogate comes in, and something like an algal model, like describing an algal model which is related to ASF. If you can start running these mitigation studies, because now you look at your biosecurity, as you said, look at what your current, should I use this mitigant in this context? There are chemical mitigants for the air. There are chemical mitigants for fomites. There are chemical mitigants for feed, because not all chemicals can be ingested by animals, right? So there's a lot of different sort of things and different strategies you can use in the set where your farm is set up, and how, or even the processing plants. Talking about just feed, right? Even when feed is brought in for processing to make meal, that needs to be considered. Transport needs to be considered. All these things, and which mitigant, and actually there's some good evidence. There's a few papers that have already shown that mitigants can actually go and destroy the virus particle. And it's that sort of data you're after, is understanding how the virus is destroyed.
Right. So the idea of, we have to understand how the virus works so that we can understand that there is no silver bullet. There is no one size fits all solution. By understanding how the virus works, how it is stable, how it moves throughout the system, that'll tell us to some extent what strategy will work in each one of those different parts you were listing. And there's probably not one strategy that's going to work for all of them.
Right. It's complex. But the good news also is, right, if you can kill something as horrible as ASF, or an ASF-like virus, right, if you can get rid of that, means you basically 99.9% there when it comes to the other less stable viruses, right? So there's actually an advantage in trying to really get to understand how to get rid of this, or how to prevent a giant virus from entering a system.
And this we're coming up on about 30 minutes here, so we'll finish on this question. Is the end game for all of these then vaccine? I mean, is that hey, all of these, at some point someone will figure out how to make a vaccine for them, and once we have the vaccine for them, we're good? Or is it not at all that simple?
Not at all that simple. I mean, we live with influenza, right?
Right.
Vaccines. We have once it becomes endemic, it's really hard to get rid of the virus, and that's why, as starting off again, I'm a viroecologist. We need to appreciate these viruses are going to be around, so we need to be able to manage it. And one of the tools is vaccines. The other tool is prevention. And in fact, my mother always says prevention is better than cure. Declan, put your hat on before you go out in the snow. It's just simple things, right? Prevention. So, try and make it so that you are secure. And the key thing is, part of that prevention is actually fully, as I say, coming back to understanding what the virus is doing. And the good news is there are new assays around. There are new assays that actually can measure the virus now in its environment. And one of the assays which we've worked on in our lab, and we are promoting more and more, is like the viability PCR assay. And by its name, it actually tells you whether that virus is present, and if it's present, is it viable or not. And that's different to a normal PCR, which just says, it's virus is present, but doesn't mean it could be degraded, right? It could be inactive. Right. And so understanding that, and understanding that, vaccines is one option, but it's not the only option. We have to have other things in place, and chemical mitigants are one of those. And I'm convinced that's part of the, and again, like you said, right, is you got to choose the context you want to apply it in. And how do you know it works? Well, look for the experiment designs. Don't only be reliant on animal assays, because the window is short. Look for mechanistic investigations that can describe inactivations. Look for information that can describe how the virus survives in its environment. If you have that, you are far better. You have better knowledge, better data to truly predict how safe your system is.
Excellent. Well, I really appreciated our conversation. We always like to end these with a few questions that are relatively the same each time we do this. And the first is, from your perspective as it would relate to folks that might be in the audience here, which is, folks in the feed milling industry, the nutrition industry, maybe some folks on the production animal veterinary side. What would be a good resource in your mind that, if they want to stay up to date on things related to, what's going on in the viral world, what's working, what's not, where the threats are, what would be a good resource for them to kind of keep track of so that they can learn more and also stay kind of current with the current issues?
Well, Adam, one of the reasons why I do podcasts is because this is real information, straight from the horse's mouth. Right. So
right.
I'm giving you information right now. So yeah, look at the social media. Social media has been really good at trying to broadcast the sort of information that's out there. But of course, it's not scientific reviewed. So whatever I said, you need to validate. So always go to scientific literature. I mean, there is no substitute for going to the key journals. You can go to my website and see what I've published on. You can see the suite of papers I've published on, for example. Always go back to the source material. There's no substitute. Yes, grasp the information you get from podcasts and social media, but going cross-validated in scientific literature. The other key thing is, if you can, attend conferences. Conferences is where the new data gets discussed and it gets tested with amongst peers, right? If you can go to, and so here in Minnesota, we have the Layman Conference, right, which is one of the big ones for swine. Join, come have a look and see what's going on. For veterinarians, there's AASV, there's and so on and so forth. Conferences allow you to, and the feed industry, Minnesota Nutrition will also have, that we regularly have a conference here on feed. And so get to where the scientists are giving their information, right? That's where you want to be.
Absolutely. All right. On kind of a different vein, anything else from a resource kind of in your professional career that might not be specific just to your field, but things that have helped you go through your career, as far as something that anybody might find interesting? We've had folks talk about things that they've done from a leadership perspective, or things that they've done from, figuring out how to do better organizational time management. Is there anything that comes to mind for you of like, this is, no matter what field I was in, this is something that I got exposed to that I'd find really impactful to people?
Oh, that's a big one, Adam. I live a relatively sheltered life, to be honest. I'm running with me in my lab. I mean, I think what I'm always challenged by is my students, whether it's undergraduate or postgraduate students. They have different life histories. And I think one of the big key things with me is listening to other opinions, and even opinions that diverge from what you are thinking. And so it challenges you to think, well, actually, how do I better design my experiments to overcome that misconception or this perceived idea that actually is not really founded in science. And so for me, it's about really what I generally like doing is going to students. There are various clubs that they have here on campus, and I go in, when I get invited, I go and listen to what they are asking. And so that's my exposure. I'm sorry, I'm a bit boring. I'm more of academic. So my circle is very much the academic sphere.
Sure. No, I think it's a good answer. In fact, I think it's our third question is usually something related to, what are things that you've seen folks in your field, some of the traits that they exhibit that have made them most successful. And actually, I think the answer you just gave is a really good example of that, is being curious and asking for challenge and seeing, how to, I think it's incredibly important in academia, but it's obviously also important no matter where you are to say, okay, this is my perspective. Let me go find somebody who doesn't automatically have my perspective and see if that changes my perspective once I hear them give theirs. Do I have new information now to potentially change my mind. And I think that's probably something we hear the term a lot today of echo chamber, right? Whether that be socially or in our own jobs, it's so easy today to live in an echo chamber where all you ever hear is the same thing that you already know, and it just reinforces that. And that's probably not a great way to grow or develop when we think about it.
Right. And changing your field. So, I worked in marine virology. I worked in honeybee virology. Now I'm working in animal medicine. Let me tell you, they're very different fields. Every single one of them. Their questions, they all have viruses in common, but their way they deal with viruses are very different. So I'm translating my virus experience of a breadth of different model systems into one. And so I come from a different direction, I guess. And so I think that's also what gives me an edge over many of my contemporaries, is that they are in that echo chamber of studying the virus in the way they've always studied it. And actually, if you go across different systems, you realize, hang on, they do something different. But yet they're trying to answer the same question. So why aren't we adopting those methods? So, yeah, I agree.
Yeah, the diversity of experience is really, really important. All right. Well, thank you very much. My guest today has been Dr. Declan Schroeder, an associate professor working in the molecular virology field at the University of Minnesota. Thanks a lot for joining us today.
No, thank you for having me.
You bet. Again, my name is Adam Farnholz, and on behalf of Wise Genetics, coming to you from North Carolina State University, thanks for listening to the Feed Science Podcast.